And after we've had 3 very interesting presentations about forests now, we're going to move on to the animals again, and we're going to hear the REVIVO organization from Slovenia talking about the return of the Mura dragon.
Hello, yes, hi. Let me share my screen. Check if it's working. So, can you see my screen?
Yes, we can see you and hear you and see your screen. All good. So you can start.
I will start with a just short introduction. So, because you skipped from very forested areas to rivers now. And so I would like to say that the Living Planet Index, which was developed by the WWF, is a first comprehensive global report on the status of migratory fish. And they find that fish are under immense threat from human-made impacts, and that we urgently need action to stop and reverse the decline in freshwater populations, which are threatened with extinction globally. And we have seen an average decline in the extent of these populations of 84% since 1970. So this is actually a very urgent matter to discuss and try to solve with different measures. And this is what we're going to try to present now. What is the status of one specific fish that we are dealing with, which are it's basically the group of sturgeons, and the species that we're focusing on is sturgeon, Acipense rutenus. So let me first introduce myself now that we've established what we want to discuss this 30 minutes. So we are a small non-governmental organization. We are called REVIVO, meaning we want to revive our rivers. And we try to bridge the gap between science and practitioners. And we're doing this through education and awareness raising. We're doing this through scientific research and monitoring activities. And we do this through implementation in the field, and not necessarily always in this order. So we are currently a group of 3 people and 2 volunteers who are working for us part-time. And the project that I want to present today, I will show later. So first, let's just see how it came to the status that we are in today. In the history, the sturgeons were mostly targeted by fishermen for their caviar, for their eggs. And the Danube River, including its Slovenian tributaries, were historically home to 6 species. And in Slovenia, some of these species regularly occurred, and some of them all year round, like the sturgeon. But on the other hand, some of them only occurred during spawning, because the large floodplain areas in Slovenia around the Mura, Drava, and the Sava rivers were ideal for their spawning as their nursery site for their young. So the specific problem in Slovenia is not so much the fishery, which is not common and also historically was not present, but in the times of Mother Teresa in the 19th century, they decided that we need a lot of space in the floodplains for growing crops and for urbanization, basically, for the expanding population. And so the Mura River especially was regulated and paved throughout its entire course through Slovenia. And so the sturlets and the sturgeons did not have the habitats anymore, and so they slowly disappeared from these rivers. The species that we are mostly interested is actually the sturlet. And so we did the comprehensive first literature review of historical sources to identify where and when the individuals of this species were last seen in Slovenian waters. And this is kind of a summary of that findings. and seeing that we had Siberian sturgeons in Slovenia. The last sighting was in the Sava River in what was the date? I don't, yeah, the date in 2016. And then we had the sighting of Rutenus in the Drava River, dating also to 2001. And we even have Acipenser rutinus, Guldenstedti in 1883. And we think that all of these species are presumably extinct from our waters. So that's why we decided to give a go on to the measures project, in which we tried to identify the locations which would be suitable habitats for sterlets or for any other sturgeon species. And we tried to find them in those locations specifically, because we thought maybe it's just that nobody was looking since 2001, for example. And maybe that's the reason why we didn't record them anymore. And the first topic that we actually wanted to do is we would take a look the entire course of the Mura River in Slovenia, and we would check and map the habitats with a number of techniques. And we would include, because we're already doing habitat mapping, it would be a shame not to also check for habitats of other migratory species. So we did not focus only on sterlets, but we also focused on other migratory species. We mapped all the habitats, but then we specifically looked for sterlets in the habitats that were most suitable for them. And through this project, we also tested different methodologies and used different or checked different habitats, so wintering, feeding, nursery, and spawning habitats specifically for sterlets. We also did some sampling on the lower Sava River, but there's only 20 kilometers of free-flowing Sava River in Slovenia, so we did not really expect any relevant results from the Sava River, and I also focus on the Mura River in this presentation. So how was the habitat mapping going in our measures project? So following the first basically Google map identification of potential habitats, we went out there with the boat, drove along the entire section of the Mura River, and first we did sampling with the sonar. So we checked the deepest parts of the river where wintering sites for sterlets and other migratory fish are supposed to be. And we did extensive scanning and then checked the results. And on basis of that research, we decided which of the sites we will then sample to check for presence of the target fish species. And as we were doing the sonar scanning, we also checked the substratum, the sediment at those sites, to see if the sediment corresponds to the requirements of our target fish species. And this survey was done, this was actually a preliminary survey in the winter of 2018 and 19. And then the next summer, we checked also for the presence of fish species. And in this second stage, we actually were focusing with different methodologies for the different key habitats. In the feeding habitats, we performed macroinvertebrate sampling, because the fish species, the sterlets and sturgeons in general, they feed a lot on macroinvertebrates, and we know which species they commonly target. So this was one of the indicators if their feed was present at the locations that we thought were potential for their feeding grounds. In addition, we set long lines because we were expecting the fish to be feeding during the summer months. And so we did long lines to check whether the fish are also present in these feeding habitats. And you can see how this was done during sampling. So kick sampling was used for macroinvertebrates, and long lines were set for species, for larger individuals, but we also used trammel nets to check for the smaller representatives of this species. In the wintering habitats, we used some of the methods were the same, while others we chose a different method. So basically, the trammel nets were also used in the wintering habitats to check for the presence of species. But in addition to that, the wintering habitats are usually slow-moving waters where also visual census was possible. So we said, okay, we will take a subsample of sites to also check visually whether there are some fish present there or not, and which are these species. And some of the results. First, let's check the results of the wintering habitat sampling on the Mura River. And these are the three graphs are aligned. So on the top one, you can see the depths that were measured. These are the maximum depths at the sites, which were later sampled. The second graph shows the results of the fixed trammel net sampling, and the bottom one shows the results of the visual census sampling. And so what we can see, there was actually not a lot of fish present. There was, altogether, there were 16 sampling sites. 12 were sampled with both. So 10 were sampled only with trammel nets. 2 were sampled with both trammel nets and visual census. And then we also identified 4 additional sampling sites to just do visual census, because visual census can be quite dangerous in areas where there's strong current, and we had to adapt those places, those locations, to the possibility of the visual census. So what these graphs actually show, that trammel nets were quite efficient, and we collected with the trammel nets, we collected 4 individual specimens, which were so 10 species were caught, and altogether 40 specimens were caught. We found Chondrostom anasus, so the common maize. We found Rutilus virgo, cactus roach, Vimba vimba, so Vimba bram, and we also caught 10 Prussian carps, Carassius gibelio. In addition, we were successful in confirming pike presence, so Esox lucius at these sites, and also Zingle, which is zingle. And it's very important freshwater species, especially from the conservation point of view. And then we also caught freshwater bram, common carp, and white bram, which is also very important for the Mura River, because it has not been present for some time, or it was not confirmed for some time in the Mura River. And so this shows that it's true that in the deeper parts, the most species were caught, and also the most individuals. And it can be seen from these results that there is kind of a lack of the deeper areas of the river with suitable habitats. Okay, next slide. So, and this is the results of the feeding habitat sampling on the Mura River. And here you can see the bottom line is on the bottom graph, you can see the distribution of the sediments, because we think that for the feeding habitats, sediments are especially important in relation to the macroinvertebrate community. The second graph is the long lines, which were used because we think that in the feeding habitats, fish are more more interested in food, so longlines could be the better solution to sample fish in these areas. And the top graph shows the macroinvertebrate community structure at these same sites. Where there is no column, it means that there were no fish identified, but the columns with white base background means that, that we did not even record that at that site for the long lines. And it's hard to see from these graphs, but basically we did not catch any sterlet, but we did catch individuals of other species, although the catch was very low. And this is also in line with our expectations because we were only testing this method. And what we can see is that amphipoda were the main, by far the main food present at these sites. And that other species, especially Ephemeroptera, were less abundant and not even present at all sites. And here I also present, as I said, we also did a similar study on the Sava River, but on a much smaller section. And these are the results. So feeding habitats on the Sava River. And we can see that at the bottom, we also have, again, the sediment distribution. In the long line section, we have the fish, and the graph is the sampling results. So the distribution of macroinvertebrates. And here it's a very clear connection with the most diverse sediments also have the most diverse macroinvertebrate structure, and unfortunately we could not confirm this diversity with fish sampling. But this is also probably due to the low amount of long lines that we were able to put here in this sampling. Okay, and if we move forward. Okay, so now to discuss a little bit the results. We saw that sampling with long lines was very unsuccessful in our case, whereas the same methods were applied downstream in the Sava River on the Croatian side, and they were quite efficient, or they were quite successful. And so our conclusion is actually that long lines are suitable method, but only in areas where the abundance of the target species is much higher than in Slovenia. We also compared this methodology with the trammel nets, which were successful at all depths, but we were limited with the trammel nets to areas with slow currents and large depths. So in areas where there were low depths, we were not successful with trammel nets. And the same as we concluded for the long lines was also concluded for the trammel nets. On the Croatian side of the Sava River, they were much more successful. And on our side, our success was limited. And we conclude that this was due to the low amount and low capacity, low density of fish in the Mura River. And basing on these results, so the comparison also with the Croatian part, we conclude that sterlet is presumably extinct in the Mura River in Slovenia and probably also in the Sava River, given that Croatian colleagues caught a lot of sterlets and we caught none in the entire survey that lasted for 2 years. And in addition, we based this conclusion on another survey that we did, and we found out through literature review that the Fisheries Research Institute was performing electrofishing along the entire Mura River in 2015. And so they also found no sterlet in the Mura River. So all these different methods finally found out not even one individual of sterlet, so we can safely say that it's gone from the Mura River. And now, given the results of the measures project, we actually want to do repopulation, and as also other policies on the European level, like the action plan for the conservation of sturgeons, the Sturgeon 2020 program, and the Pan-European action plan for sturgeons, they all call for immediate action to save the sturgeons from in the Europe or in the also Danube region. And that is why we base also our measure results and try to find the next measures and the next projects to support repopulation of the Mura River with sterlet. And these measures need to be ex situ because, as we saw, there's no more fish in the wild. We want to establish a monitoring program using telemetry, because when you have such a small number of individuals in the river, there's basically no other method that can be useful, as we saw with our measures project. We want to support habitat restoration, because on the Mura River, there's currently no dams, but the habitat is so destroyed that probably fish even don't have a place to be. And we, of course, want to educate and raise awareness about the importance of preserving habitats in our rivers. That would be all for now. There was quite limited time, so I tried to be fast and not really go into details, but I imagine that there must be quite a few questions in regard to the presentation. So thank you all for your attention, and we're open for questions. And while you're putting your questions, I would also like to present my colleague, Jan Potočnik-Erzin, who was involved with us throughout the project, and he also helped me prepare the presentation. We prepared it together, so I would like to include him in the commentary section of this presentation. Thank you.
Yeah, thank you, Polona, and hello to Jan. Yeah, very interesting project. I have to admit, I was hoping for a different outcome, not that there are no sterlets present, but we are hoping for the future. And actually, our headquarters here of the European Wilderness Society are located at the start of the Mura River. So it's a personal matter for us. So we're really hoping that we're going to get some better results in the future.
Yeah, absolutely. We're all hoping that we can bring this iconic species back, because it also impacts the entire food chain, basically, because it's a big fish and has an impact on the food chain.
Hello, everyone.
Hello, Jan. Yeah, we already have one question from Arjit. Any idea why no fish habitats were found in the Sava River? Might it be disturbance or ecological conditions that do not fit the species niche?
Yeah, absolutely. That's why we also didn't put a lot of effort on the Sava River. The Sava River, above the selected location, has 6 large hydropower dams. So the fish have no way of going upstream to the areas which were historically suitable for their spawning. And it's also below the hydropower dams, the impact of hydropicking is pretty huge. Even down in Zagreb, they get a change of water level of 1 meter in half an hour. So even the fishermen are endangered by these water level changes. So this is also why we think that there's very little fish present in that section.
Yeah, unfortunately, that's a common issue, not only in the Mura River, but all over Europe and even all over the world. Like you said, recently some very devastating results from all over the world about the populations of migratory species. So this is definitely a case that represents a worldwide problem here.
Yes, I think so. But it's we rarely see so much effort being put in one species in finding it. But yeah, once you do so much research and targetedly seek and look for one species, I think that then it's pretty safe to say that it's gone. But then this allows you to do more conservation or repopulation and habitat restoration, because we can now say that actually on the Sava River, the hydropower is there. So there's not much that we can do except working with hydropower to change their water management, release when and how do they release the water from the hydropower. This would have an immense effect. But on the other side, on the Mura River, on the Slovenian side, there's no barriers. So the Mura is the only free-flowing river left in Slovenia, and it's connected to the Danube. So given that, we expect if we improve the habitats, the fish will come back and will have somewhere to be. So our focus is on habitat restoration at the same time as we repopulate the river with sterlets.
Okay, that sounds like there's something to look forward to in the future, at least.
Yeah, absolutely.
Okay, I don't see any other questions at the moment. Yeah, so I would like to thank you again for this interesting presentation. It's really an inspiring project.